Hi, my name is Trip Oliphant, and today I'm going to be discussing two case studies that were presented, one being on pyruvate kinase and another being on a baby who is struggling with some type of glycogen issue, and we'll get to that momentarily. But first, I want to start off with pyruvate kinase and kind of how that goes along with the cell and the regulation. So the first, like I said, the first study I'll be addressing is in regards to how the enzyme pyruvate kinase is regulated within the cell and the importance of understanding the need for pyruvate kinase. So from our studies, we have learned that pyruvate kinase is important because it can be broken down or degraded to form what we know as ATP. And we also know that ATP is important because it's the cell's main energy source. The activity of pyruvate kinase is highly dependent upon activity of metabolites within the cells as well as what we know as signaling pathways. And I'll discuss those in a moment. We can see why it would be important to regulate this enzyme since we do not need it at all times, because sometimes the cell needs to use the ATP that we are actually producing. But then other times there are not enough resources available to produce more ATP. Therefore, we need some ways to regulate pyruvate kinase. So one of these ways to pyruvate kinase is regulated is it can be reduced by what is called phosphotyrosine growth signaling pathway. This reduction of pyruvate kinase is only present when the growth signaling is active within the cell. This is beneficial to the cell because it's an effective means of glucose metabolism. That includes tyrosine-mediated signaling. While phosphotyrosine does reduce the amount of pyruvate kinase, there are other means of regulation. This other means is pyruvate kinase regulation can be reduced through what we know as intracellular ROS, which is reactive oxygen species. And the way this works is it inhibits pyruvate kinase M2 through oxidation of cis-358. And when this takes place, it allows for the levels of the glucose to be shuttled to the pentose phosphate pathway, which causes for the reduction in the reactive oxygen species. When it happens is, like I said, the pentose phosphate pathway. So one other way pyruvate kinase is regulated is through the lack of nutrients for the cell, where acetylation of K3O5 causes a lower affinity for pyruvate kinase, or as we know, phosphoenol pyruvic acid. And this allows for degradation of pyruvate kinase, which eventually does reduce the concentration of pyruvate kinase within the cell. So moving on to the next part of the question, it was in regards to cancer cells and the metabolism and how that's involved with pyruvate kinase. So I'm going to touch on that now. So the key enzyme in cancer metabolism is PKM2, which we know as pyruvate kinase M2. While most cancer cells have increased expression of PKM2, in cells that are rapidly proliferating, like what we know as embryonic cells, which are constantly proliferating and growing, and adult stem cells that are always proliferating, we see what we know as MYC. So MYC increases the expression of glucose transporter 1 and also pyruvate kinase M2. This thereby promotes the Warburg effect, which we know as this effect as common with cancer cells by producing energy through a significant rate of what we know as glycolysis. And so glycolysis is happening much more rapidly, and this increases lactic acid fermentation, which actually goes to the outside into the cytosol of the cell. While my MYC is an oncogenic transcription factor, it's overexpressed in cancer cells. The MYC is associated with the promotion of what we know as RNA splicing. And RNA splicing helps with the expression of PKM2, which is pyruvate kinase M2, like I mentioned before. and that is also linked to the reason for proliferating cells. MYC is thought to induce the expression of splicing factors that favor splicing of PKM2 over PKM1. MYC stimulates glucose uptake and glycolysis, and MYC also induces glutamine consumption and metabolism, which appears to be important in glucose or oxygen-deprived conditions. So that's it for that question. I'm going to move on now to my next case, which involves a baby and glycogen. So the one I want to touch on is about a baby suffering from what I believe, after doing some research, to be glycogen storage disease. So glycogen storage disease is actually the inability to use glycogen. There were 4 options that we can choose from for diagnosis of this baby, and that was the one that I chose. And I'm kind of going to go through the steps with you on why and how I came to that conclusion. So we know that glycogen is stored glucose in the body, and this gets broken down through glycolysis to form glucose when it's low. When glucose is high, glycogen is being stored. Therefore, this baby is not able to use its glycogen storages. The reason I feel that this baby is suffering from this condition is due to the patient having some symptoms that typically occur with this disease. One of those would be an enlarged kidney and liver, which is very indicative of this disease. Another that we learned through administration of epinephrine would be high levels of lactate within the blood. And then the rest of the symptoms that we oftentimes see with the glycogen storage disease is we would usually see puberty, late puberty, and then issues with growth and impaired growth or late puberty. And that's not currently shown here because it is a very young baby, but it could potentially show up later in life. So since I've discussed the symptoms, and why I feel the baby has glycogen storage disease, I will now discuss the symptoms of the other diseases listed in the question to confirm my diagnosis. The disease, first off, galactosemia, includes loss of appetite and vomiting, and these symptoms just are not consistent with the patient that we have at hand here. The next disease I see is hereditary fructose intolerance, which involves symptoms such as nausea, gas, and abdominal pain, and another that it has is malabsorption of certain nutrients. And again, these are not consistent with with the baby's symptoms. So the final disease that we will go over is G6PD deficiency, and this includes symptoms such as anemia, fatigue, dark urine, and pain in the abdomen and back. And typically, that's not one of the symptoms that this baby is showing, so we won't typically use that as one of our disease diagnoses. So since none of the symptoms that we discussed here in the past 3 options are consistent with the clinical symptoms of the baby in question, I feel that it's very safe to assume that this baby unfortunately suffers from option C, which is glycogen storage disease. And then while there is need for further diagnostic testing to confirm the disease is likely that the baby is suffering from this illness, because I can't confirm it off of what we've done. But there is not and for treatment, there unfortunately is not exactly a treatment for glycogen storage disease, but through research I have found that through things such as diet therapy, this has been helpful for a lot of past patients that have dealt with this disease. So that's the end of my case study. I hope that it was a little bit informative, and look forward to learning more in the class.